Welcome to the deep dive. So today we're diving into the world of bacteria.
Okay.
And I know what you're thinking, bacterial. Not exactly thrilling stuff, right?
Right.
But you sent in this research on bacterial immune systems, specifically something called CRISPR.
Yeah.
And let me tell you, this is way cooler than it sounds. We're not just talking about, bacteria fighting off the common cold here.
Uh-huh.
This research, it uncovers a tool with the potential to completely change how we edit genomes.
Yeah.
And get this, including our own.
It's pretty amazing stuff. Before CRISPR, scientists, we kind of thought bacteria, they had pretty basic immune systems,
Right.
But this research, it just totally changed our understanding. It revealed the system, this incredibly adaptable and complex system. It's like finding a high-tech lab inside a tiny little bacteria.
Okay, so we're comparing CRISPR to an immune system for bacteria.
Exactly. Just like us, bacteria, they're constantly under attack, right? Viruses are trying to invade them all the time. And they've evolved this incredible defense system to fight back.
So how does it work? We talking like tiny white blood cells fighting microscopic invaders?
Not exactly. Not exactly. It's more about recognizing and remembering the enemy. Imagine this, a virus. It attacks a bacterium, but the bacterium, it survives the attack.
Okay.
So what happens is the bacterium, it takes a tiny piece of that virus's DNA, almost like a souvenir,
Okay.
and it integrates that DNA into its own genetic code.
So it's like they keep a record of all the viruses they've encountered, like a most wanted poster.
Exactly. A most wanted poster. I like that. It's like having a whole library of viral mugshots so they can recognize and neutralize these viruses if they ever show up again.
Wow. Okay, that's brilliant. But I mean, how do they actually use this information? Do they just like check the mug shots every time a new virus comes along?
You can think of it as a three-step process.
Okay.
You have the adaptation phase. That's where they get the viral mugshots. Then there's the expression and interference stage, and that's where things get really interesting.
Okay, so adaptation, that's like taking the mug shot. What of this expression and interference? What's happening there?
So this is where the bacteria use those DNA mugshots to create these short RNA segments. They're called CRAA. And these CRRNAs, they're like little detectives, each one carrying a mugshot. And they're going around looking for a match.
Oh, okay.
They team up with these special proteins called Cas proteins.
Right.
And together, they patrol the cell looking for any sign of that viral DNA.
So the cRNA is like, hold on, I've seen that DNA before. You're busted.
Exactly. And that matching, that's the interference part. They're identifying the viral DNA before it can do any damage.
Okay, so we've got the mugshot. Our detective cRNA is on the case. This is like a microscopic game of Clue. What happens next?
Well, that's where that third step comes in, remember? Destruction.
Right.
Yeah.
Destruction. What is that, like the cRNA calls in for backup?
That's a great way to put it.
The molecular SWAT team.
Exactly. And that backup is a protein, a special protein called Cas.
Cas. Okay.
And this research actually, it focuses on a particular type of Cas protein called Cas9.
Cas9. Wait a minute. Why does that sound so familiar?
Well, you've probably heard about CRISPR-Cas9 gene editing. It's kind of a big deal.
Right, right.
This research you sent, it was actually a really important early step towards that.
Hold on. Are you saying this bacterial immune system, this CRISPR thing, it's being used to actually edit genes, like in a lab?
You got it.
Wow.
And what makes it so remarkable, this research, it's not just that it uncovered this really elegant defense system in bacteria.
Yeah.
It revealed a tool, and that tool is the Cas9 protein.
Okay.
And that protein, it's capable of cutting DNA.
So Cas9, it's like molecular scissors.
A perfect analogy.
Well,
and not just any scissors, these are programmable scissors.
Okay, now that is really interesting.
The researchers, they discovered that Cas9, it needs two things to work. First, you have the cRNA, which we've talked about, and that acts as a guide, so it tells the Cas9 where to cut.
Right.
But it also needs another RNA molecule, and this one's called trachRNA.
Two RNAs. Why two? Why not just one?
Okay, so think of it this way. Imagine a pair of scissors.
Okay.
Those are your Cas9, right?
Right, yeah.
Now let's say you've got a safety latch on those scissors.
Okay.
That's the trachRNA. So it needs to be, switched off before the scissors can cut anything.
Right.
The cRNA, that's your hand, it's guiding the scissors to the exact spot on the paper you want to cut.
So the trachRNA is like the on switch for the Cas9 scissors.
Right.
And then the cRNA tells those scissors exactly where to cut. This is starting to sound less like bacterial immunity and more like a microscopic gene editing kit.
And that's what's so amazing about it.
Yeah.
They realized If we can control Cas9, well, we can potentially cut any DNA sequence we want.
Okay.
But there's one more really important detail that this research uncovered.
Let me guess. More acronyms.
Just one more, I promise. So we talked about Cas9 needing the CRNA guide to know where to cut, right?
Right.
Well, turns out there's like a safety mechanism built in.
Okay.
Cas9 also has to recognize a specific DNA sequence, and this one's called the PAM sequence.
PAM sequence.
And that sequence, it has to be right next to the target DNA before Cas9 will make the cut.
So it's like a double check
Exact.
to make sure it's cutting the right thing.
It prevents any accidental cutting of the bacteria's own DNA.
Right. You don't want those molecular scissors going rogue.
Exactly.
But how does this PAM sequence, how does that play into actually using Cas9 for, editing genes in the lab?
Well, it means that to use Cas9, you need to know the PAM sequence for whatever organism you're working with.
Wow.
It's a crucial piece of the puzzle. And that actually brings us to one of the biggest breakthroughs in this whole research.
Okay. All right. What is it?
They figured out how to combine the trach RNA and the cRNA into one single RNA molecule.
Oh, wow.
Remember that scissors analogy? It's like being able to unlock the safety latch and guide the scissors to the right spot, all in one motion.
Wow.
And this single guide RNA, it made the whole system much, much easier to work with.
So instead of two separate instructions, they basically gave Cas9 like a super instruction.
Exactly. And that was a huge turning point. Scientists suddenly had this much simpler and, more importantly, more programmable system for cutting DNA.
And I mean, with pinpoint accuracy, right?
Exactly.
So let me make sure I'm getting this right.
Yeah.
We started with like bacteria defending themselves from viruses.
Right.
And now we're talking about basically having the ability to like rewrite the code of life.
It's pretty remarkable, isn't it?
It's mind-blowing to think that this research, this tiny little thing, could completely change how we approach, well, everything.
And it really highlights why basic research is so important. You never know where the next big breakthrough is going to come from.
That's why we deep dive from the inner workings of, a bacterial immune system to the absolute cutting edge of genetic engineering.
It's an amazing journey.
It really is. And it leaves us with so many questions. I mean, if we can program Cas9 to edit any DNA sequence, what are the possibilities? What could the future hold?
The possibilities are pretty much endless.
That's incredible. Well, that's it for today's deep dive into this amazing world of CRISPR. Thanks for joining us. And don't forget to check out the show notes for some links to more info on this.